Hello and welcome back to Down the Scope. Today I wanted to have a little look at a section of an earthworm. Many of you will have seen an earthworm before and not been very impressed. From a distance they don't look like much and you might have had trouble in telling its arse from its elbow. Perhaps fewer of you will have been made to dissect an earthworm in a biology class. Earthworm is the common name for a wide variety of different species. Currently there are thought to be about 6,000 species of terrestrial earthworm. What most people think of as earthworms are those from the genus Lumbricus, but even this genus has almost 700 species in it. The most common of these is Lumbricus terrestris, and the slides I have are likely from this species. Given an earthworm, there are two simple ways to cut it. You can cut it transversely so that you end up with a circle of an earthworm, or you can cut it longitudinally, along its length, and end up with an earthworm-shaped section. In this longitudinal section we can see the basic units of earthworm anatomy, the body segment. An earthworm is composed of between 100 to 150 segments. Every segment is separated by a strand of connective tissue called the septum. However, they are not completely cut off from one another, with fluid able to pass between body segments through pores in the septum. This is a transverse section through a typical earthworm body segment. At this level of magnification, we can begin to identify several important structures. First of all, we can orientate ourselves. Just like the points of a compass, animal anatomy has its own terminology to describe what part we're talking about. Dorsal would be the equivalent of north. In vertebrates, dorsal is identified by the spinal column. In the case of earthworms, there is a large blood vessel, the dorsal blood vessel, which identifies in which direction dorsal is. Ventral is the equivalent of south, the part closest to the ground. We don't know which side is left or right, but don't worry, neither did the earthworm. In earthworms, this is identified by the ventral nerve cord and ventral vessel. So here's a quick tour of the top histological highlights of the humble earthworm in transverse section. First off, we can start from the outside. The first layer of the earthworm is its epidermis. It's formed of a single layer of cells, but if you look closely, there's a thin, non-cellular layer covering the epidermis. We can tell it's non-cellular because we can't see any cell membranes forming outlines, and there are no nuclei as there are in the cellular layer. This is the cuticle, which is colourless, transparent, and forms a protective layer over the epidermal cells. Within the epidermis you'll notice a couple of different kinds of cell. The most common is the supporting cell, which are columnar and provides structural strength to the epidermis. This is an image from a different section. It's stained slightly differently from the previous one, but demonstrates a second kind of cell quite nicely. Amongst the supporting cells there are what appear to be holes. These are goblet cells which secrete mucus that coats the worm and prevents it from drying out. They look like they have a big hole in the middle because the staining process has washed away the mucus inside it and doesn't stain it a different colour. Here we can identify a third type of cell. It looks secretory, but its contents are not translucent like the goblet cell. This is an albumin cell. They secrete something, but their function is not known. Below the epidermis there are some layers of muscle, but before we examine the muscle itself, there are a couple of other important features. Here everything looks pink, with the exception of some brown, granular material. These are pigment granules and give the worm a brownish colour. They're more abundant on the dorsal surface. I'm going to cheat and use an image from another section to illustrate the next feature. In this image, we can see blood vessels looping up through the muscle to just beneath the epidermis. You'll have noticed that worms don't have any lungs. Instead, they rely on diffusion of oxygen across their epidermis. Diffusion is a relatively slow process and only works effectively across small distances. So to get oxygen into the blood, the vessels have to loop as close as possible to the epidermis. Looking at the muscle layers, you can see two distinct sections. The difference between the two is the direction in which the muscle fibres are orientated. In the outer circular layer, the muscle fibres run across the image. In the inner longitudinal layer, they're in cross-section, so must be orientated perpendicular to the circular layer. These muscle layers work together using alternate contractions coordinated by the nervous system to help the earthworm inch along. Interrupting the muscle layer and jutting through the epidermis, we can see another organ of locomotion, the earthworm setae. These are hair-like spines which are present on every body segment except the first and the last. In this species of earthworm, there are 4 pairs of setae on each segment, but other species have different numbers and arrangements. Each setae has a muscle which is used to control its position. It can either be retracted or protracted depending on the movement of the worm. Underneath the muscle layers there is the coelomic cavity. In life it's filled with coelomic fluid, which helps with movement and structure, providing enough pressure for muscles to work against and stopping the worm from collapsing. Coelomic fluid is usually a milky white colour. There's even a species of earthworm, Earthworm fetida, whose coelomic fluid smells like garlic. When startled, many earthworm species are capable of ejecting their coelomic fluid through pores on the dorsal surface of the epidermis. One species, Didymogasta sylvaticus, is known as the squirter earthworm for its ability to shoot coelomic fluid up to 30 centimeters in response to mechanical irritation. Waste materials can build up in the coelomic fluid, so the worm has a mechanism for filtering it, just as we use our kidneys to filter waste products from our blood. The structure responsible for this is the nephridium. Each body segment, except the first 3 and the last, have 2 nephridia with 1 on either side. They are long convoluted tubes, which means that their overall structure is difficult to appreciate on a single section. The nephridium begins at the nephrostome, which pokes through the septum between body segments. Again, I'm going to use a picture from the longitudinal section to demonstrate the nephrostome. If we zoom in a bit, we can see a line of tall cells with fine hairs covering their surface. These are the cilia of the marginal cells lining the nephrostome. The cilia beat and move coelomic fluid into the mouth of the funnel-like nephrostome. From there, the fluid moves down a series of different sized tubes. We can make some of these out at a lower power. Here we can see the thinner narrow and middle tubes with the much wider muscular tube, which will ultimately lead to the outside world via a nephropore on each segment. Some of you will have noticed something odd in the muscular tube, perhaps something that shouldn't be there. This is a Rhabditis nematode, a parasite of earthworms. The larvae live in the nephridia and wait for the worm to die. Once the worm dies, they mature to adults by feeding on the bacteria in the worm's decomposing body. Sometimes they get a bit lost and insist in the body wall, and there's an example in this section. Infestations can be quite heavy, as this picture of a nephridium shows. So there you are, another of biology's curiosities, a parasitic worm within an earthworm's kidney. Moving on to the other internal organs, the most prominent feature is the intestinal tract. Just like the intestinal tract of other animals, it has a few features which attempt to maximize its surface area for absorption. The first feature is a large fold called the typhlosul. The cells which do the absorbing are the epithelial cells. If we zoom in, we can just about see the second most important adaptation for increasing surface area. The surface of the cells looks a bit fuzzy. If you use your imagination a bit, or I get a higher resolution picture, you can see tiny hair-like projections. These are microvilli, small finger-like projections of the cell membrane. They're different from the cilia we saw on the nephrostome in that they're not able to move. Instead, the membrane will be packed with proteins involved in absorption. More membrane means more proteins can fit onto the surface, which means absorption will be more efficient. The epithelial cells of the gut are long, thin cells with nuclei at the bottom. These cells also produce digestive enzymes, although there's some suggestion that symbiotic bacteria also play a role in producing enzymes which will digest the earthworm's food. Underneath the epithelial cells are 2 layers of muscle, a circular and longitudinal layer, which will contract alternately to squeeze the gut content along, much in the same way that the muscle layer underlying the epidermis helps the worm to move. The final layer of cells is composed of chlorogog or chlorogogon cells. If you see pictures of a dissected earthworm, the gut looks yellow. The chlorogog cells are responsible for this coloration. If we look at them closely, you can see that the cytoplasm is filled with tiny granules. These granules are filled with different molecules like glycogen, a glucose storage molecule, or urea from the breakdown of proteins. The chlorogog cells in earthworms are roughly equivalent to your liver. They regulate blood glucose homeostasis as well as processing waste products like urea for excretion into the coelomic cavity. That seems like plenty to be doing with today. I've not covered the nerve tissue you can see in this section, but it's probably better to discuss that in the broader context of the earthworm's nervous system, which I plan to do in another video looking at the earthworm's head, which contains exciting organs like the brain and the testicles.